In this video, we're going to look at the lysozyme mechanism as we wait for the timer to go away. There it is. All right, let's look at lysozyme. There we go. So what is lysozyme? So lysozyme is an enzyme that will destroy the bacterial cell wall or the peptidoglycan layer. And it does it by hydrolyzing the beta-1,4 glycosidic linkage, so hopefully we remember what beta-1,4 means, from N-acetylmuronic acid and N-acetylglucosamine, or what I'm going to call NAM and NAG. Lysozyme can also cleave beta-1,4 polynag, which is found in fungi, insects, and crustaceans. So lysozymes in our tears and any basically secretion that we have, and it's really one of the best understood enzyme mechanisms that we know of. That's why we're going to look at it, and we're going to be looking at it in terms of the hen egg white lysosome, because this is one of the most studied lysosomes there are, or lysozymes rather. And so here's NAG-NAM. Here we are cutting it to form our break in our bacterial cell wall. Here's an image of lysozyme and with our substrate of 6 sugars. Our important amino acids are GLU and ASP, as we will see. So let's take a look of how lysozyme binds our substrate. And it'll bind it, only binds 6 sugars. So out of the whole peptidoglycan layer, it's looking for just a 6-sugar substrate to bind on to. And we're going to label these sugars A through F. Now, sugar D, or the 4th sugar, because of the way it fits in the pocket, the ring of that sugar gets slightly distorted to be in a half-chair conformation. No other rings in this 6-sugar substrate are distorted. For lysozyme, B, D, and F are always going to be NAM because you don't have this lactyl side chain. If you look at the, especially the D and the E position, you have a lot of amino acids there that are, D and F positions. So D and F positions. You have a lot of amino acids that are interacting with the sugar, and if you had NAG there, it wouldn't be able to interact as nicely, wouldn't be able to fit. So that's why we always have B, D, and F being NAM. And we're looking to cleave between the D and the E substrate. So lysozyme is cutting right here. And D will always be the beta enantiomer by the way that this mechanism works. Remember, enzymes, the way they work is that you're always going to get one stereoisomer out. So here, we're always getting the beta isomer out. And we're showing this, so you see this 18O, that means we don't have 18 oxygens. Rather, the way that this experiment was done was we used radioactive oxygen, seeing where it went on our substrate, and we always found, oxygen's always added to the beta position here. So first, to understand how lysozyme works, we have to understand how the cutting happens without an enzyme. And so really, lysozyme is the hydrolysis of a glycoside. And really what we're doing is we're taking an acetal group and turning it into a hemiacetal. If this was to happen non-enzymatically, it is generally an acid catalyzed reaction. So an acid is going to be attacking this or being added to this oxygen, and you have an ROH leaving group. So this group would leave because you have a positive oxygen. And you're going to be forming an intermediate called an oxonium ion. That is, you're going to have a carbocation shown here that has a resonance stabilized structure where you have a double bond and you have a positive oxygen, which is incredibly rare and really energetically unfavorable. Oxygen wants to be negative, not positive, and so you are forming an oxonium ion. Then water will come in, H will leave, and you'll find a OH being added here. So an OH will bind to this carbon to make it neutral again, and you'll get your hemiacetal. Now, if you remember from the previous videos, we've said that enzymes really want to bind to our intermediate state. So we're looking for an enzyme to bind to our oxonium ion here. This is what we're trying to bind to. So that's what lysozyme's goal is going to be. So to do this, the two amino acids that are going to be doing this catalyst, this acid-base catalyst, are GLU35 and As52. And so let's just, before we look at the mechanism, let's look at these amino acids. So As52 is surrounded by a bunch of polar residues that are all conserved, and you have this long hydrogen bond network. What that means is that As52's pKa is generally unchanged, and this As is going to be stabilizing our oxonium ion. Remember, oxonium is positive. You have a negative charge As here. We're going to have an interaction for stabilization. Glue, on the other hand, is in its protonated form, which we're not used to seeing. Usually we think of glue as a negative amino acid. The reason why GLU35, not 3.5, why GLU35 is protonated is because it is surrounded by nonpolar amino acids. Having a negative charge in a nonpolar environment is very energetically unfavorable. Therefore, the pKa of glutamic acid skyrockets. It goes to a pKa that is similar to histidine. Therefore, in the pocket, it is going to be protonated. This is going to be our acid. This is going to be donating that hydrogen. All right, with our two amino acid players now introduced, let's look at this catalyst. So step one is we have to bind our substrate, our 6 amino acids, and we're going to be focusing on the D and the E sugars. When binding, D is going to be contorted to that half chair conformation. So that's step one. Step two, glue is going to act like an acid. It's going to transfer that proton to oxygen 1 that is between the D and the E ring. This cleaves our carbon-oxygen 1 bond, forming your oxonium ion. This oxonium ion, remember, is positively charged, so we have asp stabilizing our substrate right here. The E ring, it has an OH on it, the H coming from glue. It no longer has this glycosidic bond, so it leaves. So we have chopped our peptidoglycan layer already. But before we're done, we have to restore our enzyme and take care of this oxonium ion. So step 3, As52 is going to act like a nucleophile. Remember, it's donating electrons, and it's going to attack this positive carbon. And you're going to have a covalent bond, covalent catalyst. So that's step 2. Step 3, so step 2 is happening right here. We have an intermediate where we're bound to our enzyme. Step 3, or rather step 4, water comes in. It's going to go where the E ring was, and it's going to do two things. One, we need to restore our glue to our positive charge, or sorry, our protonated form. So glue will take the hydrogen from water. The water will attack the D ring. That will cut the covalent bond from asp in the D ring, which means we have restored our enzyme, and this D ring has an OH, so it leaves. This is a base catalysis now. We have restored our enzyme, and lysozyme is ready to chop another peptidoglycan layer. And that is the mechanism of lysozyme. That ends this video. I will see you in the next video.